Bismillahirrahmanirrahim. Today's lecture is related with sense of taste. The stimuli that we know as taste or flavor are actually mixture of 5 elementary taste qualities: salt, sweet, sour, bitter, and yummy. Taste stimuli that are particularly effective in eliciting these sensations are respectively sodium chloride, sucrose, hydrochloric acid, quinine, which is used for the treatment of malaria, and obtained Synconia plant, and monosodium glutamate, which is present in tomato and cheese. Yummy has been described as a proteinaceous meaty flavor. In general cooking, meat flavor develops as a result of interaction between amino acid, peptide, reducing sugar, vitamin, and nucleotide or their breakdown product during cooking from the lean components. Taste receptor. The sensation of taste depends on the activation of chemoreceptors located in the taste bulb. Here you can see this is the taste bulb, and the taste receptor are located in this taste bud. A taste bud consists of a group of 50 to 150 chemoreceptor cells and as well as supporting cell and basal cells. These are the basal cells, these are the chemoreceptor cells, these are the supporting cells, and these are the basal cells. The chemoreceptor cell synapse at their bases with primary afferent neuron. Here you can see that these chemoreceptor cells make a synapse with the afferent nerve fiber. Type of chemoreceptor cells. There are two types of chemoreceptor cell, can be distinguished by difference in their synaptic vesicle content. These are the vesicle and its content. They can differentiate by the content of their vesicle. One type has dense core vesicle, whereas the other has clear round vesicle. In this you can see that the vesicles are dense and the other they are clear and round. The apices of the cell have microvilli that extends towards a taste pore. Here you can see that these are the microvilli which extends to a depression which is called the taste pore. Lifespan of chemoreceptor cells. Chemoreceptors cells live only about 10 days. They are continually replaced by new chemoreceptor cells that differentiate from the basal cell located near the base of the taste bud. These are the basal cell, and these chemoreceptor cells are differentiated basal cells. The location of each type of taste papillae. The tongue is divided into two portions or two parts, the anterior 2/3 and the posterior 1/3, by a V-shaped sulcus which is called circumvallent sulcus. The taste buds are located on the tip of the tongue. Taste buds are for the sweet. On the side of the tip of tongue, the taste buds are for the salty taste, and at the upper quadrant on the side of the tongue, the receptors are for the sour, and the posterior 1/3 on the surface of the tongue in that area receptors are present for the better test. These are the different receptors. The fungiform papilla, which look like a fungus, the falciform papilla, which look like an antenna, and palate papilla, which look like a leaf, and circumvallate papilla, which is covered by the walls like the fetus in the uterus. Now the fungiform papilla are actually for the sweet and yummy taste, and palate papilla are for the sour taste, and circumvallate papilla are for the bitter taste. Chemoreceptor cell is specialized for one type of taste stimuli set on the microvilli of chemoreceptor cell. These are the microvilli, and on these microvilli you can see that these receptors are present and detect stimulatory molecule that diffuse into the taste pore from the overlying fluid layer. When we chew the food, the taste molecule dissolve in that fluid in saliva, and from there the taste molecule actually enter the taste receptor. When salty foods are ingested, the sodium from the food enter the taste cell via open epithelial sodium channel. On the epithelium, the sodium channel are always open, and when we take salty food, that salt enters through these open channel into the receptor cell. The resulting depolarization open voltage gets sodium and calcium channel, leading to release of serotonin onto the apparent taste axon. These are, here you can see that when sodium enter, it open the sodium channel and the calcium channel, and the calcium enter into the taste receptor and act on the vesicle and rupture them. These vesicles release their neurotransmitter on the apparent taste axon, and in this way impulses are sent to the higher centers. Food tastes sour because of their acidity, and when acids are present in water, they produce hydrogen ion protons. Hydrogen ions are also called protons. Proton enter cell through an ion channel, and they block potassium channels. These channels for proton are present on the villi, and they are open, and when the food which contain hydrogen, that hydrogen enter into the chemoreceptor cell and block the potassium channel and prevent the efflux of potassium from the receptor cells. The decreased efflux of potassium along with the presence of the proton depolarize the cell, causing voltage-gated sodium and calcium channel to open. This hydrogen or proton cause the sodium and potassium channel open, and there is influx of calcium and sodium inside the chemoreceptor cell, and the calcium act with the vesicle which release its neurotransmitter, and in this way the signals are sent to the higher center like salt taste transduction. Bitter, sweet and yummy compound all activate taste receptor cell via G protein coupled receptor. The bitter receptor are from the T2R family, taste receptor type 2 member family of receptor protein. Humans have over 25 such receptors, which are important to avoid ingestion of dangerous substances like poison and toxins. Activation of the G protein receptor use a second messenger system to increase intracellular calcium, which open ion channel, allowing the influx of sodium. These ion channels depolarize the cell and cause ATP specific channel to open, allow ATP to enter and synapse and act on the apprentice axon and produce action potential and send signals to the higher center. Here you can see this is the G coupling protein. When it is activated, the alpha component is detached and it produces a second messenger which is called phospholipase or IP3. This IP3 act on the endoplasmic reticulum and open the calcium channel, and calcium is released from the endoplasmic reticulum to the intracellular fluid, and it also opens the sodium channel. This calcium causes activation of the ATP channels and open them, and ATP is released on the apprentice axons, and in this way better signals are sent to the higher centers. Sweet and yummy receptor are comprise a G protein couple dimer, meaning two separate protein functioning together as one. There are two protein for sweet taste and they act together as a one protein. The receptors are encoded by the T1R family of receptor protein. Sweet receptor are of the T1R2 and T1R3 protein. T1R2 means taste receptor type 1 member 2 and taste receptor type 1 member 3 proteins. Both proteins need to be present and functioning for activation of sweet taste cell. Like beta cell, activation of the G protein receptor use a second messenger system to release calcium from the intracellular store and increase the influx of sodium. Here you can see that G protein is activated and second messenger is produced, phospholipase C IP3, which act on the endoplasmic reticulum and release calcium from it to the interior of the chemoreceptor, which act on the ATP channels, open them and release the ATP to the apparent neuron and produce signals which are carried to the higher center. The ion channel depolarize the cell and cause ATP specific channel to open, allow ATP to enter the synapse and act on the apprentice neuron. Yummy receptors are comprised of the T1R3 protein like the sweet receptor but is paired with the T1R1 protein. Once the G protein coupled receptor is activated, the transduction pathway is same as bitter and sweet taste. Here you can see that it is the same pathway as per the sweet and bitter. Taste pathway. The tongue is innervated by 3 cranial nerves. The front 2/3 of the tongue is innervated by cranial nerve 7, which is called facial nerve. The back third is innervated by cranial nerve 9, glossopharyngeal nerve. Finally, the epiglottis and pharynx are innervated by cranial nerve 10, which is called vagus nerve. Here you can see that this is the anterior 2/3 of the tongue, which is innervated by the facial nerve, and the posterior 1/3 is innervated by the 9th, the glossopharyngeal and the epiglottis, and pharynx is innervated by the 10th nerve, the vagus nerve. They carry a sensation from these areas. All 3 cranial nerves enter brainstem at the medulla and synapse in the nucleus of the solitary tract. You can see these all 3 nerves then enter into the medulla, and here is solitary tract and the synapse there. From there, information is sent to the posterior medial nucleus of the thalamus. This is ventral posterior medial nucleus of the thalamus. So this tract, which originates from the tractus solitary, they send impulses to the ventral posterior and medial nucleus of the thalamus. Thalamic neurons send projection to the gustatory cortex. From here you can see that then projection are sent to the gustatory cortex. The gustatory cortex is located deeply in the lateral fissure in a region called the insula. This region is called the insula. This is the gustatory cortex and it is located here, and this area is called the insula. Information processing stays primarily on the ipsilateral side of the nervous system. This is the taste sensation which are sent to the higher center, the gustatory center. They are located ipsilaterally. Initially they are located ipsilaterally, but projection within the brain also exists between the taste region and the hypothalamus and amygdala. Here the projections are also from the ascending tract of the taste projection are also sent to the hypothalamus and amygdala, which is part of the limbic or nucleus of the limbic system. So this is story of the pathway. Here you can see that from tongue the 7th, 9th and 10th cranial nerve receive information and they are sent to the solitary tract, which is located in the brainstem. Solitary, they are sent to the ventral posterior medial nucleus. This is the ventral posterior medial nucleus, which is located in the thalamus, and from the thalamus signals are then sent to the primary gustatory cortex, which is border of frontal and temporal lobe cerebral cortex. This is the frontal lobe and this is the temporal lobe, and it is make a border between these two lobes. So this was sensation and its pathway. Thank you. Please subscribe the channel and press the bell icon for upcoming notification. Thank you once again.